We've been talking about association areas. We talked about visual and auditory association areas. Let's continue with auditory association areas and talk about aphasia. Aphasia refers to loss of language ability due to brain injury. So there are many, many forms of aphasia, all right? This is a very complex topic. We're just going to skim the surface here. Just want you to have a basic idea of what aphasia is and maybe a couple little variants of it. Often results from stroke, trauma, could be brain tumor. One way or another, those auditory areas of the brain are in some way damaged. Now, Broca's aphasia is also called non-fluent aphasia, results from damage to Broca's area. So those are Brodmann areas 44 and 45. Notice those are in the frontal lobe. These are the areas that control the production of speech. So the ability for me right now to think about something and then to make the words actually come out, that's Broca's area that's doing that, all right? So in Broca's aphasia, all right, individuals can understand language, but they have difficulty speaking it. So some of you may know Gabby Giffords, our former congressional representative, wasn't she? Or House of Representatives? Congressional? I don't remember, I'm sorry. Senator. But she was shot right here in Tucson at the Safeway down there at Ina Oracle. Or is it a Fry's? I don't know. But she was shot through the left side of her head, and it damaged her Broca's area, and she had Broca's aphasia. She had to resign from Congress. She couldn't carry out her duties anymore, and it took her quite a while. She now, I see her in commercials, she now speaks, but it's clearly with difficulty. And right after, she didn't appear in public for quite some time because she simply couldn't speak. They did a lot of work with her, and what an incredibly courageous and heroic woman, what an amazing person. But she had Broca's aphasia. She had great difficulty speaking, and she's gotten much better through therapy. Depending on the particular type of Broca's aphasia, there may also be difficulty in writing, typing, or using sign language. And here's where it gets really, really cool and interesting and unusual. That sometimes someone won't be able to speak, but they can still sign. Or sometimes they can't speak or sign, but they can type. And, whatever. So it just kind of depends on the exact nature of the injury. But in one way or another, Broca's aphasia means you have trouble either speaking or writing or typing or using sign language. In other words, it's the production of language. That's where the difficulty is. In contrast, Wernicke's aphasia, fluent aphasia, results from damage to Wernicke's area. Brodmann areas 22, 39, and 40. So you can see those. We talked about those before. Those are over there on the temporal and part back on the parietal lobe. This is where individuals cannot understand language and they speak nonsense. So what happens, they call it fluent aphasia because people are speaking, but they're not really saying sensible things. So, think about it. In Broca's aphasia, you can say something to someone and they'll understand you, but they simply can't produce speech back. They may have very limited or even no ability to respond to you with words. In Wernicke's aphasia, people hear you talking, but they can't really make any sense out of what you're saying, and then when they speak, it likewise comes out just nonsense. So, you'll say something to them, they'll look right at you and go, "Ababin glabadibri il sizin bajalubidin," and you're thinking, no, I did that yesterday. But there are, again, varying degrees of this, all right? Sometimes they'll speak actual words, but they'll put together in nonsensical ways, sometimes described as word salad, or it's just incomprehensible. So poor people, either one, the aphasia, oh my gosh, how difficult would that be? Because language, we kind of take language for granted, but it is such an essential part of our lives. So aphasia, difficulty, loss of language ability. Broca's and Wernicke's are the two most common types. Actually, there are areas that connect those two. There are parts of the brain, tracts that connect Broca's and Wernicke's, and you can damage those as well. So there's altogether, there's like 17 different kinds of aphasia. As I said, we're really just barely skimming the surface, okay? But I want you to have a basic understanding of Broca's versus Wernicke's, okay? many other kinds involving related Brodmann areas. Damage to corresponding areas in the right hemisphere affect body language, including tone of voice and gestures. So sometimes following stroke, for example, if those corresponding Brodmann areas are damaged on the right side, people still use language okay, but they lack any expression in their voice, all right? And sometimes they look really stiff and stuff. Turns out that, body language, we have, when we're talking to people, we use movements with our hands, with our facial expressions, and so on. Damage to the contralateral hemisphere can cause the ability to still use language, but the loss of sort of body language, facial expressions, gestures, and so on. So, very interesting stuff, all right? Notice 2 million people in the United States suffer from aphasia. That's a lot. And fortunately, there are really good therapies. Interestingly, sometimes I've heard what they do is people with damage on the left side to Broca's and Wernicke's, the language areas, parts of the right side of the brain are involved more with singing. So sometimes they'll teach people to sing the words rather than speak them. And that actually can help them regain some of their abilities. Continuing with association areas, the prefrontal cortex, the association area, prefrontal association area, Brodmann areas 9, 10, 11, and 12. So look out there, that's the very, very front of the frontal lobe, okay? This is probably the most complex and sophisticated of all association areas. It's like, the question is almost not what does the prefrontal cortex do, it's almost like what doesn't the prefrontal cortex do. Numerous connections to other association areas, including the thalamus, the hypothalamus, the limbic system, cerebellum. This is involved in all the stuff I've been talking all the way along about the frontal lobe being involved in this. It's really up to now I've been saying the frontal lobe. I've really been meaning the prefrontal cortex. It's just we didn't know about the prefrontal cortex yet, so I wasn't saying that. But reasoning, learning, memory, personality, judgment, conscious, intuition, mood, planning, initiative, formation of abstract ideas, all right? Damage to these areas produces profound effects, even dramatic changes in personality, and other just, I don't know how to describe it, just like weird stuff. Like, for example, let's say somebody gave you a deck of cards, and they said sort it by suit. You would make piles for, clubs, diamonds, hearts, spades. Or they could say sort it by number. You would make a pile for the aces, for the twos, the threes, the fours, all the way up to jacks, the queens. Or they might say sort it by color. You would sort it into the reds and the blacks. People with damage to prefrontal cortex, you can give them a deck of cards. I mean, again, it depends on specific damage. I'm just giving one example of the kind of thing that can happen. Someone with prefrontal cortex damage, you give them a deck of cards and you say sort it by suit, and they'll start sorting by suit, but partway through they'll switch to sorting by color, and then they'll switch to sorting by number, and you end up with piles that are just completely all jumbled together. That's the kind of thing that damage to the prefrontal cortex can do. An inability to plan, an inability to figure things out, as well as dramatic changes in your mood and your judgment. People make bad decisions after damage to that area, formation of abstract ideas. This is the most complex part of the brain, and so not surprisingly, damage to this area produces really, really profound effects. Damage, for example, to the frontal lobe or prefrontal cortex may result in what they call dysexecutive syndrome. Dysexecutive syndrome includes impaired movement, because remember the motor cortex was there, impaired language because Broca's area is there, depression affecting the mood, lack of empathy. So other people will tell you about a horrible thing that happened and you just don't show any emotion. Loss of inhibition with respect to emotions, excitement, and anger. So inappropriate emotions, laughing hysterically when something horrible happens, stuff like that. Social inhibition. So in some cases people become completely withdrawn and shy and won't interact. Repetitive movements, inappropriate use of objects, compulsive eating. By the way, the social stuff can go in the opposite way. If you've taken an intro psych class, you may have heard of Phineas Gage, the railroad worker that the big metal spike got blown through his head, and his personality basically completely changed afterwards. The unfortunate part of that story is that they usually don't follow up. As it turns out, he pretty much went back to normal again a few years later. So a lot of the damage seemed to be mostly temporary. But nonetheless, it did affect him throughout his whole life. You can't just have a whole giant chunk of your brain blown out and completely go back to normal. But the stuff you've heard described about Phineas Gage, the fact that he used to be a responsible worker, a foreman, and so on, and afterwards he took to drinking, and he was always late for work or whatever. Some of that really was true, even if it did fix itself later on. But that's dysexecutive syndrome, a complete change in your personality and your moods and all that kind of stuff. Prefrontal cortex, holy smoke, damage to that area, you can become really like a completely different person. We've been talking about the cortex, all the white matter, the gray matter rather. Let's start talking about the white matter, the underlying white matter. So again, in the upper right, you see that the gray is the cell bodies, the ganglia, the dendrites, and so on. You can see how that's pretty much all out around the edges. The white matter is more in the core of the brain. Remember, white matter, myelinated axons, that's all the wiring. So out around the edges are where all the cell processing is taking place, and the white matter is basically just the wires that are connecting the cells together. Cerebral white matter, myelinated interneurons. All right. Association fibers connect different parts of the same hemisphere. So, we need our vision input, our auditory input, our thinking, our emotional input. They all affect one another, and so different parts of the cortex have to tell each other what's going on. This is how they do that, by what are called association fibers. That's the white matter, the myelinated axons that are allowing different parts of the brain to talk to each other. Projection fibers connect the cerebrum to other parts of the brain. So association fibers connecting parts of the cortex together. Projection fibers are the ones that are radiating out to other parts of the brain and the nervous system. Consists primarily of the corona radiata and the internal capsule. Right? Then there are also commissural fibers. They connect the two hemispheres together. The main one are the corpus callosum, and then also the anterior and posterior commissures, which we've seen in the lab. So in that diagram on the lower right, you see the commissural fibers. The diagram on the lower left are a lot of the projection fibers. So again, three basic categories of fibers. Association are connecting different parts of the cerebral cortex, the gray matter. Projection are allowing the cerebrum to talk to the brainstem and the cerebellum and so on. And then commissural are simply allowing the left and right hemisphere to keep track of what's going on. Each side needs to know. And here you see the corpus callosum, 250, I've seen 300, different estimates, doesn't really matter. 250 to 300 million myelinated axons that are simply allowing the left and right half of your brain to know what the other one is doing. That's very important. Okay, let's talk about the basal nuclei. For many years, the basal nuclei were called the basal ganglia. And notice why they should be called basal nuclei. Remember, ganglia are clusters of cell bodies out in the peripheral nervous system, whereas nuclei are collections of cell bodies in the central nervous system. So in the brain, they should be called basal nuclei. I'll warn you, though, that many old textbooks and sources will still call them the basal ganglia. Just get used to it. It's not anything to be upset about. Just deal with it. It's a basal because they're down at the base of the forebrain and on the top of the midbrain. So look in the lower left and lower right. Lower left, I think, a good example there. You can see, there's the thalamus, and then coming out you see the putamen and the caudate head, the caudate body, the caudate tail, the nucleus accumbens. And then again on the lower right, you can see all these clusters, all these different area clusters. See how they're at the base of the cerebral cortex, of the entire cerebrum, but they're kind of like on top of the midbrain. So that's why they're called the basal nuclei. They're deep down. These are deep structures, deep down inside of the brain. The lentiform nuclei consist of the putamen and the globus pallidus. Lentiform means shaped like a lens. And putamen has the root word puta, which you may know. The root word means shell, and that's how it gets its other vulgar name as well, because it kind of looks like a shell, doesn't it? So putamen, globus pallidus, the pale globe. So what happens here is, if you can look in the bottom right, you can see the globus pallidus, and you can see also there the putamen. Notice how they're sort of lens-shaped. They're curved like the lenses on glasses. That's why they're called lentiform nuclei. They have a curved lens-like shape. Okay? The corpus striatum, literally the striped body, consists of the lentiform nucleus and also the caudate nucleus. Okay? And that's where in the lower left you see the caudate nucleus. You can see the caudate head, the caudate body, the caudate tail. All right, so just a little basic anatomy there so you can see where they are. These structures are functionally linked to the subthalamic nuclei and to the substantia nigra. Remember the substantia nigra, the black stuff, those were the eyebrows of the monkey in the midbrain. That's where we have the dopaminergic neurons that when they died, that death led to Parkinson's disease, all right? The basal nuclei get input from the entire cortex and connect to the prefrontal and premotor cortices. So therefore, they are involved in the connections between thought and sensation and movement. Remember, they're also connecting to the midbrain as well. So very, very complex relationships here that all have to do with your thoughts and your emotions and your movements and your thinking. So they heavily influence movement. They don't directly control it. Remember, the most immediate control of movement came from the precentral gyrus, from the motor cortex. So, but these are all influencing the motor cortex. They're taking all kinds of other inputs, again, from memories and emotions and from sensory stuff and like that, and they are influencing. They're slightly, they're modifying what the precentral gyrus and the basic motor cortex do. So continuing with basal nuclei, they overlap functionally with the cerebellum, because again, remember, the cerebellum is what's involved in smoothing, making movements smooth and fluid, and also controlling balance and posture. So altogether, the basal nuclei then have a major role in starting and stopping movement, all right? They kind of like give the go-ahead to actually start moving, and then they give the go-ahead to go ahead and stop movement, and they also monitor the intensity of your movements. And they're involved in controlling what we call stereotyped movement and in inhibiting unnecessary movement. So again, things like, stereotyped movement, when you walk and you swing your arms, all right, that kind of a thing. Relatively involuntary, very stereotyped, inhibit unnecessary movement, so they kind of provide a limitation on movement so that you don't, reach way past something. So imagine what happens then if there's damage to the basal nuclei. Fundamental things like arm swing end up getting affected. Ability to precisely control your movements gets affected, all right? You start making unnecessary or exaggerated movements. You have trouble starting movement and stopping movement. This is classic behavior in Parkinson's patients. They stand up and they want to start walking, and you'll see them sort of shuffling their feet. They sometimes call it the Parkinson's shuffle. They're trying to start walking, but they can't. They can't get the movement started. That's so characteristic of damage to the basal nuclei. Basal nuclei receive somatosensory input from the cerebral cortex, from the thalamus, and from the brainstem. Integration takes place in the corpus striatum, and then signals will be sent back to the input structures via feedback loops. So, very important again, this is all feedback. You're monitoring your movement as it's happening, and then you're having feedback loops to try to adjust, to try to compensate, to try to smooth things out and make it all work in a fluid way. So output through the globus pallidus to the motor areas of the cerebral cortex, and plays a role in the selection then of the smoothing of movements. All right, so look at how all this works together. Again, the brain is so incredibly complex. We've got the midbrain, the cerebellum, the cerebral cortex, and the basal nuclei all working together to try to ensure that we have natural, smooth, coordinated, appropriate movements. And you can think of the basal nuclei kind of as being at the very center of all that. And so that's why damage to the basal nuclei just makes everything go haywire. Movements just don't work the way they're supposed to anymore. So dysfunction of the basal nuclei can lead to a variety of disorders, including Parkinson's disease, Huntington's disease, Tourette's disorder, which you may have heard of before, and also obsessive compulsive disorders. Again, think about the influence on, as we said, stereotyped movements. It's so complicated because, again, the inputs are coming from all over the cortex. They're in some way modified by our memories and our emotions and so on. So really the basal nuclei at the very core of all this kind of stuff. And so when something goes wrong there, holy smoke, you have really profound effects on movements. So zooming in just a little bit here, Huntington's disease is an inherited disorder of the corpus striatum, which again was part of that corpus striatum, the striped bodies, the caudate nucleus. Loss of GABA neurons in this case leads to overactivity of dopamine neurons. So GABA, remember, the inhibitory neurotransmitter in the brain, it was one that was sort of slowing things down. It was decreasing activity. So when you lose GABA neurons, that means everything is sort of like constantly overexcited. So you get overactivity of the dopamine neurons that are actually causing the movement. So onset, unfortunately, typically comes between the ages of 30 and 50. So you don't know when you're young that this is going to happen to you. And because it's genetic, it means that if your parents had it, there's a good chance you'll get it. I forget the actual probability, and I can't believe I've forgotten that. I do know that. It slipped out of my mind at this very moment. It's characterized by what they call chorea. It used to be called Huntington's chorea. Chorea, a Greek root word meaning dance. So what happens here with people with Huntington's disease is they're constantly sort of flailing their arms and jerking their legs and stuff like that. It's as though they're doing like a horribly uncoordinated dance. So, and again, that's caused by damage to the GABA neurons. Those GABA neurons normally should be inhibiting movements, but because they're damaged, movements are now sort of out of control. These quick, jerky, non-repetitive movements. That's what happens in Huntington's disease. Oftentimes people actually have to be restrained so they don't hurt themselves or other people. I mean, they will suddenly like throw their arm out, and if there's a table right there, they could smack their arm, they break their arm on the table or something. It's a very, very sad disorder. Woody Guthrie, you may have heard of Woody Guthrie. I don't know if they talk about him in elementary school anymore. This land is my land, this land is your land, this land is my land. It's just somebody's land, whoever it was. Woody Guthrie, he had Huntington's chorea, all right? And eventually you get cognitive impairment leading to dementia. Again, it's very sad. This is a very sad disorder. The drugs can treat it a little bit, but it's in House, one of the main characters, one of the main doctors, found out that she had Huntington's disease. That was one of the main themes in the show. Parkinson's, we've talked about a few times, results from loss of dopamine neurons in substantia nigra. So the dopamine are the movement neurons. So in a way, you sort of get the opposite of Huntington's. Now the probabilities of Parkinson's patients have trouble moving, all right? Exact cause is unknown, appears to be both genetic and environmental. Onset between 45 and 50. Parkinson's, there's a classic Parkinson's posture, a stooped posture, little arm swing, partially flexed arms. Look in the lower right there. That's a classic. I'll bet you've seen old people kind of hunched over like that, shuffling along. They call it the Parkinson's shuffle. What you get is some tremor, bradykinesia, which is slow movement, and difficulty initiating movements. Like I mentioned before, Parkinson's patients, they stand up, they want to start walking, they have trouble getting that first step, all right? They have trouble initiating the movement. So here you go. Disorders of the basal nuclei. I was saying before just how they're kind of at the center of all the movement-related stuff. So as you can see, man, damage in the basal nuclei, and you can get some really profound movement disorders. Famous people with Parkinson's, a little twerp on the left, Michael J. Fox, actually a great character. And of course on the right, Muhammad Ali, float like a butterfly, sting like a bee, hands can't hear what eyes can't see. Both of these guys became ambassadors for Parkinson's disease. They were very open about their Parkinson's. They talked about it, and Muhammad Ali already did. Michael J. Fox actually has been a real ambassador for awareness for Parkinson's disease, making people more aware, funding treatment of it, stuff like that. He's been a real benefit to the entire Parkinson's community, and people make some jokes about him, which he laughs about because he's a cool guy. That's what happens when he tries to take a selfie. Great at making martinis, terrible at stealing tambourines. He laughs about this because he is a great guy. Michael J. Fox is truly a great guy who has brought a lot of attention to Parkinson's, and there's his signature. Horrible humor, but he laughs at it. He is a great man. Let's talk about some other things left over. We've really covered the cerebrum and the different parts of the brain, the brain stem, the diencephalon, the cerebrum. Let's talk about some other topics that are kind of sidelines, but they're at the same time essential to everything. So cerebrospinal fluid, we've been mentioning it and talking about it for quite some time. Let's kind of zero in and make it more specific here. A clear, watery fluid found in the brain and the spinal cord. That is cerebrospinal fluid. Cerebrospinal. It's in the brain and in the spinal cord. Filtered from the blood by the ependymal cells in the choroid plexuses of the ventricles. So remember we had the blood-brain barrier, which was going to keep things out of the CSF. So the CFS, we have the different glial cells. Remember all the kinds of glial cells? So ependymal cells were the specific glial cells found in the choroid plexuses in the ventricles. That's where we're actually creating the cerebrospinal fluid, and then we're controlling what's in the cerebrospinal fluid via the blood-brain barrier. Enters the subarachnoid space in the fourth ventricle. So we've been looking at this in lab. Down in the fourth ventricle, that's where it gets into the subarachnoid space, flows through the subarachnoid space and the central canal of the spinal cord. So those are the two places that it circulates, through the central canal of the spinal cord and also in the subarachnoid space. Remember, that's why we had the denticulate ligaments. Those were designed to keep the subarachnoid space open, to keep it patent, so that there can be an easy flow of CSF through that space. It's recycled by arachnoid villi, sometimes called arachnoid granules, from the subarachnoid space into the dural venous sinuses. So you can see there on the top diagram the superior sagittal sinus, and in Bio 202 we learned there's also an inferior sagittal sinus. There's the cavernous sinus. Those are all places where we can recycle cerebrospinal fluid into the CSF. So it's being made in the ependymal cells and then recycled into the dural venous sinuses. So there's a constant recycling, a constant flow. So we constantly keep cleaning up the old CSF, getting the waste products out, and then making brand new CSF, and then letting in through the blood-brain barrier all of the glucose and the oxygen and all that kind of stuff. It provides buoyancy to the brain, so your brain basically floats in cerebrospinal fluid. It helps as a shock absorber, helps minimize cerebral trauma, cushions the brain to protect it from trauma, provides nutrients to the cells, all right? So amino acids and glucose all circulate in the CSF, and clears the waste products, as I was just saying. And that's all due to the blood-brain barrier. We don't let just anything in. We let in the nutrients and things that are supposed to be there, and don't let in all the bad stuff. So the ventricles themselves, let's just specify and be explicit on this. 4 interconnected cavities in the brain that contain cerebrospinal fluid. This is where it flows. They contain the choroid plexuses. Plexus, root word means a tangle. So we have nerve plexuses. We also have plexuses of blood vessels. So it just means a whole bunch of blood vessels all kind of tangled together. In Bio 202, we'll see other plexuses as well. Here, plexus just means this big group of tangled blood vessels up in the ventricles, and that's where there are the ependymal cells that are making the cerebrospinal fluid. 2 lateral ventricles, one in each cerebral hemisphere. We don't number them as 1 or 2, neither left nor right gets a preference. But you can see them, they're kind of like big C clamps. They're shaped like big C clamps. They have the different parts, they're different horns, they're called. Third ventricle is between the 2 halves of the thalamus. So notice in that neat rotating diagram up on the top. So the blue are the lateral ventricles. The yellow is the third ventricle. And see how there's a big hole in the third ventricle? That's where the intermediate mass of the thalamus goes. Remember the thalamus were the 2 big egg-shaped structures? So the thalamus is in fact sort of underneath those top horns on the lateral ventricles. See how the thalamus would kind of fit in there? You could have like a big egg. So on both sides of that yellow thing, imagine there's a big egg sitting on each side, and that egg will kind of fit underneath the lateral ventricles. That's where the thalamus is, all right? And then the fourth ventricle is in between the pons and the cerebellum, and then that's the purple or the magenta or whatever. And then there is the red connector, that is the cerebral aqueduct, okay? Remember that runs right through the midbrain, that's the mouth of the monkey. That's surrounded by the periaqueductal gray matter, okay? Again, it's very difficult with the brain to try to keep imagining this in 3 dimensions. I try to keep reminding you of where other structures are to try to help you visualize that. It's difficult to do. Then the interventricular foramina connect the lateral ventricles to the third ventricle. So see, in between the lateral ventricles and that yellow third ventricle, there are those little sky blue tubes. Those are the interventricular foramina. This is how, therefore, notice, all the ventricles are interconnected. So cerebrospinal fluid can flow freely through all the ventricles. Okay, it's constantly circulating through all of them. CSF flows from the lateral ventricles into the third, down the cerebral aqueduct into the fourth, then into the central canal and into the subarachnoid space. So constantly flowing, constantly being recycled. That's where the nutrients and the waste products of your brain are carried.